MMP seminar series. I would like to begin by acknowledging the Wurundjeri people of the Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you all are joining me from today. I pay my respect to their elders, past, present, and emerging, and to the Aboriginal elders of the other communities. Hi, I'm Ria. I'm one of the PhD students from the University of Melbourne, North of the Centre. Before we begin the presentation, I would like to remind everyone that the seminar will be recorded and it will be available in our CCMB website. Also, please make sure that you keep yourself muted during the end of presentation and wait till the end of the presentation to ask your questions. You can also put your questions in the chat box and we will read them out during the Q&A session at the end. Now, I would like to introduce you to our speaker today, Dr. Raphael Trenker. Dr. Trenker is a postdoc fellow in the lab of Dr. Natalia Jura at the University of California, San Francisco. Dr. Trenker obtained his PhD in 2018 from the University of Melbourne, where he joined with Drs. Matthew and Melissa Call in the Structural Biology Division at VHI. During his PhD, he investigated transmembrane helix-helix interactions in single-span transmembrane receptors using crystallography and deep mutational scanning analysis. He then joined Dr. Natalia Jura's lab at UCSF in 2018, where in collaboration with the group of Dr. Klimen Verba, he applies Cryo-EM to investigate structural basis for the activation mechanism of human epidermal growth factor receptors. For today's seminar, Dr. Trenker will be discussing about cryo-EM structures of active HER2-HER3 receptor complex and how these structures reveal the dynamics at the dimerization interface, which is induced by the binding of a single ligand. Dr. Trenker, a warm welcome to you, and we can begin whenever you're ready.
Okay. Thank you. Thank you very much for having me and giving me the opportunity to talk here about this project. As you said, this is a joint effort between Natalia Jura's lab at UCSF and also Klim Verba, who I think is a really wizard and taught me basically everything I've learned in the last 3 years about cryo-EM. So very grateful for that. And thank you for having me. I'll be talking about our structures that were recently published on the HER2-HER3 neuregulin beta signaling complex. And HER2 and HER3 are both human epidermal growth factor receptors. And also feel free to interrupt me anytime if you like. But yeah, so both receptors are human epidermal growth factor receptors of the receptor tyrosine kinase family, which means that these receptors are primarily expressed and present at the plasma membrane. And there they encounter extracellular growth factors that then leads to receptor dimerization into active signaling complexes. So ligand binding events are here translated directly into tyrosine kinase phosphorylation, and then kicks off various signaling pathways that tend to inhibit apoptosis and deliver pro-survival signals. And it also can lead to proliferation, migration, and differentiation, depending on the signal and the receptor complexes that are being activated. There are a total of 4 HER receptors in the family, and they all share this extracellular ligand binding domain. They have a single transmembrane alpha helix and an intracellular tyrosine kinase domain that has this C-terminal tail attached, and this C-terminal tail is the main phosphorylation sites through these kinases and serves as the docking site for various downstream signaling proteins. Now, there's these 4 HER receptors, and quite remarkably, these 4 HER receptors are activated and stimulated by over a dozen different ligand families that we don't actually know a whole lot about. And these different ligands can induce various hormone heterodimers of these HER receptors. And what we are interested in figuring out in the lab and as parts of some of the project that I work on is how these different ligands can induce specific HER receptor complexes and lead to ligand-specific downstream signaling and activation of various pathways. So we're very much interested in studying mainly structures of all of these receptors and try and figure out what they all mean. But 2 receptors here are particularly interesting from a medical and biochemical point of view. And these are these 2 here, the HER2 receptor, which is interesting because it can't be activated just by itself, because HER2 does not have a known growth factor that can bind to it and activate it. And HER3 is also super interesting because HER3 can be bound by a ligand, but it has this inactive pseudokinase domain. So it can't catalyze tyrosine phosphorylation itself. However, when HER2 and HER3 are co-expressed and present in the same cell, and they're activated or bind to neuregulin 1 as their ligands, then HER2 and HER3 can assemble into this heterodimeric, very potent signaling complex. And in this signaling complex, HER3 is the ligand binding unit, and HER2 basically provides the active kinase to the signaling unit. Because unlike other or many other kinases, her receptor kinases are not just activated by phosphorylation of the kinase itself. These kinases are activated by allosteric stimulation of one kinase of the other. Here, this is a structure of the HER3 pseudokinase domain together with the EGFR kinase. We do not have any structures of the HER2-HER3 kinase complex, but this came out of our lab a few years ago. And what we basically see here and in other HER receptor kinase dimers is that one kinase here, in this case, the inactive HER3 pseudokinase, which can sort of activate the HER2 kinase by pushing the alpha C helix in to this catalytic cleft here and keeping EGFR in this case, but we think this works similar in HER2 in an active conformation. So even though HER3 is catalytically inactive itself, it can partake in an active signaling complex by allosteric activation of the EGFR HER2 kinase in this case. At least we think that. And once HER2 is activated, this then allows HER2 to catalyze phosphorylation reactions. And this goes via activation of RAS GTP down the MAP kinase pathway. And HER3 in particular is a very strong activator of the PI3 kinase pathway because it has 6 docking sites for this protein. And this is one of the most mutated pathways that we know in cancer. So this signaling complex is very potent because of in particular PI3 kinase activation. And it's biochemically interesting, but therefore also medically very relevant. HER2 in particular is overexpressed in about 20 to 30% of breast cancers. And we also know of a variety of missense mutations that occur both in HER2 and HER3, and they really happen anywhere in the receptor. But the most frequent one is this mutation that occurs in the HER2 extracellular domain. And this is super interesting because HER2 is an orphan receptor, it doesn't bind a ligand. And before we had the structure, we really had no idea how such mutation in the extracellular domain can cause disease and have activating effect on this complex. And as I mentioned before, we don't, until recently, we basically had no structural understanding or no structures of the HER2-HER3 heterocomplex itself. And so it was very important for us to get some insights there. And I've introduced you how we think the kinase domains are activated and how they come together. There are no structures of the extracellular domain for especially the HER3 protein, but we do know how activation of the HER receptor family might occur because of various studies done on EGFR, the founding member of the family, basically. So when EGFR was crystallized now about 2 decades ago in an inactive conformation, which means not bound to a ligand, it exists in the so-called tethered conformation, which is characterized by this beta hairpin protrusion that I'm showing here in magenta that is called the dimerization arm. And this dimerization arm is sort of hidden away in the structure. And what happens now upon ligand binding, in this case EGF, ligand basically binds between domain 3 and domain 1 here. And this induces a very large conformational change of the EGFR receptor. And this conformational change then exposes this dimerization arm here. And with this dimerization arm exposed, 2 EGFR receptors can come together and form this, I think, very beautiful heart-shaped extracellular domain dimer that is, when EGF is bound, perfectly symmetric. And what I want you to take away from this here is that this is just, remember this heart-shaped extracellular domain structure, and that this beta hairpin protrusion, the dimerization arm, leads from one receptor deep into this pocket on the other receptor. So it sort of reaches over and grabs onto the dimerization partner. And it is this dimerization arm that provides most of the interaction surface between these 2 receptors. There's some additional interactions happening here, but most of the energy really comes from dimerizing through these dimerization arms, which is why they're called dimerization arm. And we also know that the way these dimerization arms are positioned and holding these structures together is very important for downstream signaling. Because a couple of years ago, Mark Lemon's group obtained structures of the EGF receptor bound to different ligands, EGF and epiregulin. And what these ligands do is induce very slight conformational differences. Some people might not pick up on this or think this is not a very drastic change, but this asymmetric structure now induces a very different signaling outcome in the same cell type than EGF stimulation does. EGF leads to proliferation, epiregulin to differentiation. And this basically shows us that small differences in the way these dimerization arms come together and regulate the extracellular domain structure is very important in the signaling outcome. So when we now look at the dimerization arms of our receptors of interest, HER2 and HER3, something very curious has been observed now some time ago. When we look at structures of the HER receptors, we find for EGFR, HER3, and HER4 that without the ligand, they exist in this tethered conformation. However, HER2 was crystallized or has always been crystallized in this already active-like extended conformation that is characterized by this dimerization arm being freely exposed. And this sort of led to this long-standing mystery or the long-standing question as to why then, if HER2 has this dimerization arm exposed, does not undergo homodimerization and activation by bringing the two HER2 kinases together. And really didn't have a good understanding or a good idea of why this is. Some groups suggest that this conformation of HER2, even though it looks like an active conformation, is actually auto-inhibited by certain other interactions. And to truly know what this conformation of HER2 means, we needed to understand whether HER2, when it undergoes the transition from being a monomeric inactive receptor that is not able to signal, if this assembles into a heterodimeric active signaling complex, whether HER2 undergoes an additional conformational change from here into here. And if it doesn't do that, then why not? So we really wanted to obtain a structure of the HER2-HER3 heterocomplex to answer this long-standing mystery about HER2 autoinhibition. Then we know that HER2 can't bind a ligand. So far, we didn't, or before we had the structure, we didn't also really know whether HER2 and HER3 would actually assemble into a heart-shaped extracellular domain complex, because we've never seen a structure of HER2 extracellular domain dimers that are singly liganded. And a structure we expected would possibly give us some good ideas and indications how various oncogenic mutations work and cause disease that are present in HER2 and HER3 receptors. And we figured by being able to isolate a HER2-HER3 heterocomplex, we could also address questions like whether therapeutic antibodies that are currently used in the clinic every day to treat patients with HER2-positive breast cancers, whether these therapeutic antibodies that are designed to target HER2 alone, whether they can also target and bind to a heterocomplex of HER2 with HER3. And that's very interesting from a therapeutic point of view, because we don't really know whether these antibodies compete with HER3 and, or whether they can actually be accommodated in this HER2-HER3 heterocomplex. So these are sort of the questions that we had before starting this project. The major challenges why it took very long to obtain structural information of HER2-HER3 heterodimers is because people tend to work with isolated domains, and as membrane structural biologists, I think everybody can understand why. But if you take the HER2 and HER3 extracellular domains and even the kinase domains, there's no measurable affinity for one another. So we figured, therefore, that there must be a considerable contribution of interactions between the transmembrane domain helices and the intracellular kinase domains to hold this complex together. So we wondered whether we could assemble this HER2-HER3 heterocomplex bound to a ligand using full-length receptors. And this, and I'm preaching to the choir probably, comes with its own unique challenges because we're working with full-length transmembrane proteins now. And that has challenges in expression, purification, and structural analysis itself. And what I want to do now is give a little bit of more insights how we actually got to isolate these proteins and obtain structures of them. So the overall strategy that we employ to isolate these receptors, and this has been sort of a little bit of a game changer for the Receptor project in our lab in the first place, is by purifying ligands that can activate HER2 and 3 various HER receptors and immobilizing them on flag beads. And then we can use these house-made ligand-bound flag beads to pull out HER receptors. In this case, if we co-express HER2 and HER3 mammalian cells, we can use these beads to first fish out HER3 and then select for the heterodimer by specifically doing a second pull down that specifically targets HER2. And in this way, we should be able to isolate a ligand-bound HER2-HER3 heterodimer. But before I get to this purification, I want to spend just a minute on talking about the ligands. I think since we published this, the ligand plasmid has been the most thought after plasmids for any of this, because it's not always trivial to express extracellular ligands in E. coli, simply because they tend to be heavily disulfide bonded EGF-like domains. So all HER receptor ligands have 3 conserved disulfide bonds, and expressing this in E. coli can often lead to inclusion body formation and misfolded protein. But there's this very great E. coli cell line called E. coli origami, and this cell line has a few mutations that make the cytoplasm of this E. coli strain less reducing, so that you can actually form disulfide bonded proteins. And this has been very useful for us in making all sorts of different ligands, even beyond EGF-like domains. So we can fuse this EGF-like domain, in this case of neuregulin 1 beta, which is what binds to the receptor, fuse it to this thyroidoxin, which also helps supposedly to form these disulfide bonds. And by doing so, out of E. coli, we get beautifully folded neuregulin 1 beta ligands. And just out of a 6-liter E. coli prep, we get 5 to 20 milligrams of proteins, which really lasts us for a month and is a few days of work. So this has really helped us in being able to isolate this complex, because we can form the HER2-HER3 heterodimer already straight after lysis, rather than doing this later after purifying individual proteins. And we just published a very detailed protocol, so please have a look if you're interested, and reach out anytime if you're interested in making disulfide linked extracellular ligands for your receptors. The HER2 and HER3 receptors we don't express in E. coli. Here we use mammalian cells. And another game-changing moment in this project was when we transitioned from adherent 293 adherent cultures to these XP293 suspension cultures, because these XP293 suspension cultures can be they can be grown at very high cell density. We usually do a transient transfection at 3 to 4 million cells per milliliter, and we usually get away with a few 30 milliliter cultures. So we do this transient transfection, and then the next day, as described in this protocol, you just add expression enhancers. There's 2 of them, and what we do, and what I would like to suggest for everybody to try, is we add HER2 kinase inhibitors that stabilize the HER2 kinase domain. And by adding these inhibitors straight to the expression cultures, we can actually boost our yields a lot, and we also get more homogeneous protein. And then after just another day, we usually harvest our cells and then lyse these XP293 cultures in good old DDM, and keep this also usually around for the entire purification. We screened various other ones, other detergents. Some work really well, some work not so well, but DDM seemed for us to be the way to go here. And then after doing so, we either co-express HER2 or 3 in the same cells, or we express them separately and then mix the lysates after DDM lysis. And then if we then do the ligand pull down and another pull down on HER2, we're able to get these perfectly stoichiometric complexes of HER2 and HER3 as shown on this SDS page. So this was really great to see that we could isolate this complex. However, using the wild type HER2 and HER3 receptors, we struggle to obtain enough protein to do anything meaningful with. So we did some additional engineering, and we already know that there are mutations. These are oncogenic mutations that also are observed in the clinic. And these oncogenic mutations in the HER3 pseudokinase domain, we already know stabilize interactions of HER3 with other kinase domains such as EGFR and also HER2. And by introducing these kinase domain mutations, we were able to significantly increase our yields of the full-length or near full-length heterocomplex. We chop off the C-terminal tails a bit usually. And with this, we were able to obtain enough protein to eventually actually get a signal by size exclusion chromatography. And we then also were able to obtain our first negative stain to the classes. And we were very excited to see this because here we actually had for the first time proof that the heterodimer can form these hard-shaped extracellular domains. And then there's some additional density for transmembrane and intracellular domains as well here by negative stain. So this was really cool. And what I want to talk about for a minute, this size exclusion chromatography is what I'm showing here on the y-axis, not a relative signal where we have one here. This is the actual milli-AU absorbance. So this signal that we get out of here, out of 120 milliliter mammalian expression culture is really, really low. And before this project, I probably would have just thrown this in the bin and moved on to other things. But I'm glad we didn't. If we take this, we can concentrate it down to about 0.1 micromolar receptor and 50 microliters, which is great to freeze a whole bunch of grids. But initial trials in freezing these complexes on holy carbon EM grids were not really successful. We never saw any particles because I think the concentration is just really, really low. And if anything, everything would just precipitate around the carbon. And what really saved us, and I think the reason we have a structure is probably simply because we got our hands on these very great graphene oxide-coated grids that were initially provided to us by the AGARD lab here at UCSF, which pioneered a lot of the graphene oxide geogrid work. And I want to just spend a couple of minutes talking about them because I think they were really key for us to get these structures. And if you suffer from purifying proteins at low concentrations, then I think these grids could really help everybody because you can just make them in an hour at home in the lab. So basically what these graphene oxide coated grids are, we use normal quantifoil holy carbon gold grids usually. And then we apply this very thin single layer or hopefully a single layer of graphene oxide on top. And what this layer then does is it provides a support layer for the particles to settle on. So you usually don't have this issue that all the particles that you do have precipitate on the carbon. You get away with negative stain EM-like concentration, and I'll talk about this in just a minute. And what it also often does it change the way the particles freeze, and you can reduce orientation bias. And it's just another sort of grid to simply try and see if you get any good particles on your EM grid. And these grids are also very good, and this is another thing that was pioneered by the AGARD lab, and there are published protocols out there. But what's great about graphene oxide is that you can chemically modify it. So the AGARD lab has had lots of luck adding PEG chains to make everything a little bit more hydrophilic. You can also add charged groups on top of these graphene oxide coated grids. It's really just a different way of screening things. And what's also, I think, really impressive is that you can add affinity tags onto these graphene oxide coated grids. For example, a spy catcher spy tag combination. If you add a spy tag to this graphene oxide, you can express your protein of interest with a spy catcher and just use the lysate really and purify your protein on the grid, which is really, really cool. Or you can use this covalent tag to further concentrate your protein on the grid by loading it, washing it off, and doing the same thing again until you get a good enough concentration. And I said that we are currently making these in-house. You can buy them. Supposedly, they're often not as good and not very reliable. But all you really need is this small petri dish here, you fill this up with water all the way to the top, and you need a copper mesh to put your glow discharged gold grids on. And then you just apply a single layer, or like a particular layer, hopefully a single film of graphene oxide that's solubilized in methanol on top. And then by simply pumping out this water, you can reduce the film so that it goes on top of the grids that you have here on the copper mesh. And then hopefully you get a single nice layer of graphene oxide. And this takes about an hour to do and can save your projects. So this is really great. And to get a bit of an idea of what concentrations we're talking about, if you know how to obtain good density by negative stain EM, a good rule of thumb for these GO grids is using a concentration that's about 5 to 10-fold higher of what you usually use by negative stain EM. So we usually calibrate, because these were always too low to obtain any valuable reading by nanodrop. We sort of calibrated our gel filtration column signal to what looks good on a negative stain EM grid. And then 5 times that, you get a pretty decent bare GO grid micrograph with good particle density. So it's really not a lot of protein that you need for this. And we just freeze these proteins in DDM for now. Just another practical tip, if you are working with GO grids or are planning to work with GO grids, when you use detergent solubilized proteins, we made the observation that about 40% of our data collections gave us empty micrographs. And this is because probably the DDM or the detergent washes off some of these graphene oxides areas. So what you want to do is really spend an additional 30 minutes when you set up your data collection and be really picky about where you pick your targets. And what you see here in this medium bag montage, this is just one grid square on our grids. What looks a bit like dirt is basically the graphene oxide coating, and an ideal area to image that you get the most out of your data collection would be to image here, where you clearly see some sort of homogeneous layers of graphene oxide. You can image here, I usually avoid it, but if you image here, you'll just get empty micrographs. So it's worth spending a bit of additional time in picking your targets manually. And then just as a reminder, so on this GO grid, we froze this HER2-HER3 heterocomplex bound to neuroglin-1 beta as a ligand. And when we analyzed these data, we saw very quickly in our first 2D class averages. And I hope I've shown you enough cartoons and structures of her receptor extracellular domains that you see what's happening here. What we basically see in these 2D class averages are heart-shaped extracellular domain dimers. And where we expected the transmembrane domains to be and the kinase domains to be, we see that the signal smears out, probably because these receptors are fairly flexible and every processing software just focuses on the very bulky extracellular domains. And then, to no surprise, if we ask for 3D volumes here, and generally we start processing all these data in CryoSpark, we obtained a pretty good resolution here, but we obtained almost no density at the transmembrane and kinase domains. So we had very nice extracellular domain densities with no density for transmembrane and kinase domains. And we wondered for a long time where these kinases went. We tried a lot of things. For instance, also, we wondered whether we could stabilize or reduce the flexibility by non-specific cross-linking of these receptor complexes. We usually now do that for most of our complexes in 0.2% glutaraldehyde for about 40 minutes. And we did not obtain significantly more density for transmembrane and kinase domains. But what cross-linking usually does to our receptors is that it increases the resolution that we get for the extracellular domain. And that's what I'm going to focus on for now. By doing that and focusing basically all of our processing efforts now in the extracellular domain only, we were able to obtain this beautiful structure of the HER2-HER3 heterocomplex that now showed for the first time HER3 being in an extended conformation bound to a ligand. And we also saw for the first time what we were after, and this is HER2 being complexed in an active signaling complex together with a co-receptor. And one of the very first things we did is we took our HER2 structure of the heterodimer and overlaid it to a whole bunch of other structures that we had of HER2 that came from Cryo-EM, that came from crystallography. And we were, to be honest, somewhat surprised to see that our structure overlaid almost perfectly with all these other structures obtained by these different methods. And what this indicated, though, and I think it's an important finding, is that from its monomeric inactive state into a sampling into an active complex with the co-receptor, HER2 does not appear to undergo an additional conformational change, meaning that HER2, as it is here and with the structure that we know, represents the active ready to dimerize signaling unit of HER2. And since this is the ready to dimerize signal structure of the extracellular domain, this really poses the question again, how then is HER2 prevented from undergoing autoactivation? And I think that our structure has some very important insights into why this may be. And to answer this, we need to take a closer look at this structure. As expected, we have HER2 here shown on the left in blue, and the dimerization arm of HER2 is reaching over into this pocket into HER3 to stabilize this complex. But we were very surprised to see that when you flip the structure around and you search for the HER3 dimerization arm, the HER3 dimerization arm is missing, and it's pretty much completely unresolved. And this indicated that the HER3 dimerization arm is flexible and is not required to hold the HER2-HER3 heterodimer together, which was very surprising to us because this is not what we've seen in other HER receptor structures. And we think that the reason for this disengagement of the HER3 dimerization arm is due to an allosteric connection between ligand binding and dimerization arm stabilization. If you look at the HER3 part of our structure, then you see that what ligand binding does here is it brings this domain and this domain here closer together, and this can then form a pocket, and the dimerization of HER2 can bind to this pocket and is nicely stabilized and stabilize the whole structure. In red, we can see all the interacting residues. But in contrast, HER2 does not bind the ligands, and therefore this conformational change does not occur. HER2 doesn't have such a pocket to stabilize the dimerization arm, and therefore the HER3 dimerization arm is likely only very loosely, if at all, associated with this HER2 open pocket. And if we think about this, we now know that the EGFR homodimer that was very easy, or fairly simple, I guess, to isolate and to crystallize. EGFR bound to EGF forms such a strong extracellular domain dimer because we have two ligands bound to the extracellular domain. Two ligands bound means we can stabilize and engage two dimerization arm. Therefore, the interaction surface between these extracellular domains is really, really large, and we get strong homodimers. In contrast, we learned for HER2-HER3 heterodimers, we only have one ligand bound, and therefore we only get one stabilizing pocket, and therefore we can only stabilize one dimerization arm, and we get about half the interaction surface. And if you now allow me to extrapolate this a little bit, then, and we think about possible putative HER2 homodimers, then we hypothesize that these HER2 homodimers are probably very unstable because without any ligand bounds, we cannot engage and stabilize this extracellular homodimer via the dimerization arms. So even though there's a dimerization arm present in both of these HER2 extracellular domains, they cannot be engaged in a stabilizing pocket, and therefore the HER2 homodimer might be too unstable to exist, at least under physiological expression levels. Whether they form in cancer when HER2 is overexpressed, we don't know. We don't have any evidence that HER2 can form hard-shaped homodimers, but we think that under at least normal levels, this homodimer will not form. So the biggest takeaway, I guess, from our HER2-HER3 heterodimer is that it forms a surprisingly weak extracellular domain with only one dimerization arm engaged. And then this immediately posed the question to us, well, can we somehow artificially stabilize this weak heterodimer? And then maybe nature has already done this, or cancer has already done this and stabilized this very important signaling complex. So we went back and looked into patient data. And as I mentioned earlier, one of the most frequent oncogenic missense mutations that is observed in HER2 in cancer patients in HER2 is this S310F mutation. By far, there's some other ones in the kinase domain, but this is a really puzzling one. And now that we had the structure at hand, we mapped this mutation onto our wild-type structure of HER2 and HER3. We were pretty amazed to see that this mutation appears to be located where we expected the HER3 dimerization arm to be. So we wondered whether what this mutation could be doing is stabilizing the complex by stabilizing the dimerization arm of HER3. So we made the mutation and we isolated the complex. We noticed that we can also isolate more of the complex because it's just more stable. We saw that the structure is overall remarkably similar or pretty much identical, with the exception that now the HER3 dimerization arm, as hypothesized, is perfectly visible and is as strong in density as the HER2 dimerization arm was in this structure and also our wild-type structure. And this is probably because this new phenylalanine can undergo pi stacking interactions with this conserved tyrosine that's present on the HER3 dimerization arm. This then allows formation of various other hydrogen bonds and sort of just glues and sticks this HER3 dimerization arm into this open pocket of HER2. So this sort of suggests that this oncogenic mutation is oncogenic because it stabilizes the whole HER2-HER3 heterocomplex. And we can even confirm and saw this simply by getting much better yields and purifying this. So now that we are able to, or that we were able to isolate HER2-HER3 heterocomplexes, wild-type and mutant, we were perfectly positioned basically to answer another long-standing question. And this is whether these therapeutic antibodies that I mentioned a little bit earlier that are used to treat HER2-positive cancers, whether they can bind to HER2 only, which is what they were designed to do, or whether they can actually be accommodated in a HER2-HER3 heterocomplex. And first of all, we looked at existing structures. There were structures also recently solved by Cryo-EM of HER2 bound to two of these therapeutic antibodies, pertuzumab and trastuzumab, also called Herceptin. They bind to very distinct spots on HER2. And we could then overlay our structure via HER2 on top of this structure and see that we were expecting clashes between HER3 and both of these therapeutic antibodies. Pertuzumab has very strong clashes over here in the dimerization arm region, and there's some clashes of HER3 with trastuzumab here as well. But we had everything we needed to do the experiments. We expressed some trastuzumab and pertuzumab Fab in E. coli, we purified the HER2-HER3 heterocomplexes and then incubated those with an excess of trastuzumab and pertuzumab. Did another IP after that, simply to find that the therapeutic antibodies are bound to HER2 without losing any HER3. So we could not compete off HER3. And all of this was bound together, indicating that a complex of HER2-HER3 with these therapeutic antibodies can form. It was a little bit different for the oncogenic mutation, where pertuzumab, which binds to the dimerization arm region, could not bind this heterocomplex. This is either because this interface is so strong that pertuzumab can't get in, or simply because the epitope is now too different for pertuzumab to recognize HER2. Nonetheless, this is not what we were expecting, and we were interested in following up on the conformational changes that have to occur to accommodate these therapeutic antibodies. And we were at least to obtain another structure of the heterocomplex bound to pertuzumab Fab. And I really like the structure. I think it's super beautiful with trastuzumab sticking out of HER2 down here. And if you do a little bit of overlaying, you find that what basically happens is that the therapeutic antibody binds to HER2. It rotates out a little bit here at this domain 4 where the antibody binds, and it sort of kicks HER3 out of the way. So consistent with what we learned so far, the heterodimer is flexible and dynamic enough to allow binding of these therapeutic antibodies, which has various therapeutic implications. But what we're now very interested to do is trying to use our structure and leverage our ability to isolate these complexes to figure out next whether we can design and find some bispecific binders that bind to HER2 and HER3 at the same time. And this is, I think, a very important goal in the project, because what tends to happen in the clinic is that many patients that undergo treatment with these therapeutic antibodies have to stop treatment because of cross-reactivity with healthy heart tissues. And this is because HER2 is not only expressed in breast cancer, but it's also highly expressed in the heart. And we think that the relevant complex in the heart to maintain heart function and contractility of the heart is one of HER2 in complex with HER4. It also requires neuregulin 1 beta as the ligand. And what we really want to do is be able to specifically inhibit this complex and leave this complex intact, or maybe even stabilize this complex without stabilizing this one using other antibodies or reagents. So what we aim to do here is to see whether we could isolate a similar heterodimer of HER2, HER4, just like the HER2, HER3 heterodimer. And we use lots of different strategies, and I'm not going to go much into detail here, but we can isolate and get structures of this heterodimer as well. And now we are leveraging us being able to isolate these complexes to do screenings for bispecific binders that recognize one but not the other. So this is something that we're now actively pursuing in various different ways. And another interesting part of this project, now that we're able to, how you got our feet wet a little bit on isolating HER receptor, full-length HER receptors, is to ask the question whether other HER receptors that bind to various different ligands, how they can transmit ligand-specific signaling cascades. And for here, we are sort of interested in understanding more about the underdog receptor HER4 in the family, because it actually has lots of important functions in the adult brain, also during development, and some cancers appear to be driven partially by HER4. And HER4 is biochemically very interesting because it binds to 7 different ligand families. And most of these ligands we don't really know a lot about. We do know that there is some sort of biased agonism through HER4 signaling. And we want to understand whether there's a structural basis for this biased agonism and whether there's coupling of the ligand binding domain with specific ligands to specific ways of activating the intracellular kinases. And where I'm going with this is that I really want to make sure that everybody understands that we really need full-length structures where we can see the extracellular domains and the intracellular kinase domains and transmembrane domains at the same time. And this has really been, for the receptor tyrosine kinase cryo-EM field, has been a real challenge so far. There's 20 receptor tyrosine kinases out there, and most of them are very important for human health. For instance, I'm sure many people are familiar with the insulin receptor in some ways. And every time also other labs have worked with full-length receptor tyrosine kinases by cryo-EM, what we ended up getting are just extracellular domain structures, and the transmembrane kinase domains seem to never be resolved in these structures. So this is really something that we have to figure out. And the way we've been going about this, because we think it's partially probably a processing issue. We've done a lot of 3D classification with our particle stacks and rely on, here I'm showing these 3 volumes. We get in different classes, different amount or different densities in transmembrane kinase domains. We also figured out that we can improve this by recentering our particles in the extracted box, because usually CryoSpark and Relion puts the extracellular domains right in the middle of this box, and then the signals where we want the kinase domains to be smears out. And by recentering this, we are usually able to obtain a little bit more density for transmembrane and kinase regions, or what we think are transmembrane kinase regions. And refining doesn't make this a whole lot better, but at least we end up with cryo-EM maps that are large enough to accommodate all of these functional domains. And we're working very hard on getting this processing a little bit better. It might just be a numbers game that we need really, really large data sets to capture these different states, and this might just all be very flexible. And this is my last slide before you can go into questions. But what we're also aiming to do is put these receptors, and this works for some HER receptors, into artificial membranes. We've seen some of these in nanodiscs, and I think it's quite remarkable how well you can see these kinases here sticking out of these nanodiscs on the other side. And we're in the middle of figuring out how to image these properly. And hopefully we will get some higher resolution of the full receptor complexes. And before we get into questions, I want to thank our labs, Natalia's and Klim's lab. I want to thank Natalia and Klim, and especially Klim for teaching me a lot of cryo-EM, and to Natalia for being a super supportive mentor in every other aspect. Thank you to the lab, and thanks to Devon, who is the co-author on this project. And it's been really great working together over the years. And with that, I'm happy to take any questions.
Yeah, thank you, Dr. Trenker. That was a really great talk, really informative. Now it's time for our Q&A session. Those who have questions, please raise the hand function or put your questions in the chat box. Yeah, we have our first question. Josh, would you like to unmute yourself and ask your question?
Hi, Raph. Great talk. Thanks for sharing all that, methodology. I think the, improvements you made all the way along from expression to processing made a lot of sense, and as a result of very nice structures. So my question is about the kinase domain, which is the final frontier for this, right? That is very challenging. How important do you think the phosphorylation state of that kinase area is? And did the mutations that I may have missed it, some mutations you introduced in HER2 to improve your yield, did that affect the kinase activity?
So the mutations that we made are, so the mutations in HER3, yes, so there's various indications that the phosphorylation state changes. We have some data sets where we dephosphorylate our receptors, and then, we activate them and phosphorylate them. This doesn't really seem to help in resolving any kinase domains. I do think it's probably important to inhibit our kinases in some way. So we express everything in the presence of panertinib, which is a kinase inhibitor for HER2. So the complexes that we ended up looking at the end are not phosphorylated, and they're also not active because the HER2 kinase is active. And by Western blot, after having these kinase inhibitors around, we don't see any phosphorylation. But yeah, maybe it's worth going back and looking at more phosphorylated species in more detail. But I suspect it's a processing and flexibility issue overall, and that we just have to restrict the flexibility of the complexes a little bit, or a lot.
Yeah.
But yeah, that's a good question. It's a lot to look at for sure.
Yeah, Isabel?
Yeah, I think, it's holy grail to get this, Raph, and so following on Joe's question, so you were very successful with identifying patient mutation that stabilize the ectodomain structure. And so can you apply the same principle? And there's already structure of the intracellular domain of the HER2-HER3 complexes. So can you derive some important interaction you could use to stabilize that interaction between the kinase and pseudokinase domain? And that's one part of the question. The second is the role of the PIP2 in the membrane. So is a direct interaction of the kinase domain, pseudokinase domain to the membrane surface that will really impact the stability of those complexes?
Yes, very good questions. So what we use to stabilize the entire complex is it's an EG mutation that occurs in the HER3 pseudokinase domain. We have a crystal structure of HER3 with EGFR that shows that this basically increases the affinity of we know that this increases heterodimerization. And without this EG mutation in the HER3 kinase, we're not able to obtain a lot of heterocomplex. So even though we have this stabilizing mutation in the kinase domain, we still don't see it. I assume that it forms and that they're just relatively moving together. And this is the strongest activating mutation that we know we can put into HER3 to activate HER2. But yeah, very good point. We can screen this a little bit further. We're thinking also about cross-linking the kinases because we should be able to predict where the interface is, right, by overlay. And then second part of your question was the lipid interactions. Almost certainly, her receptor kinases interact with lipids. There's a lot of modeling done on EGFR, and it's also been observed biochemically in various ways. So we're trying to put HER2, HER3 into nanodiscs. Currently, our yields are still so low that it's very challenging to get that. It works better for some other HER receptors. And we've also incorporated some PIP2 in these nanodiscs. So we're very eager to see these. We are having issues once the receptor is in a nanodisc, we can see these nanodiscs really well, but we're having issues concentrating the sample, and we're having issues seeing the particles on our grids, which is what we're currently optimizing. We're probably going to go to the SpyTex bycatcher system on the GO grids to trap the nanodiscs so we can look at them. Yeah, it's a very good question. There's an EGFR full-length structure in nanodiscs that didn't seem to help very much, but I don't think they put any PIP2 in, and I think PIP2 is definitely the one that we want to look at.
Yeah, right.
Thank you.
And absolutely great talk, Raph, and thanks for sharing all those tips. Is really applicable to many other receptor tyrosine kinases, so that's great. Thanks.
Thanks. I hope it can help someone.
Yeah, we do have some questions in the chat box. Panu, do you want to unmute yourself and ask your question?
Yes, I can ask. Thanks for that. Can you hear me?
Yes.
Yeah, I have actually two questions. One is that the area you have mentioned of the graphene oxide grids is like that looks much thicker than the normal, like the single or multi, like 2-layer of graphene oxide. So do you see that if there are any like diffraction pattern you have collected to see that what's the number of graphene oxide layers you have, or does that affect any B factor or the scattering of electrons like CTF after that? That's one question. So you like, can I ask the second question?
Yes, the other one. I'll write it down.
The second question is like you have mentioned the cross-linking as well. So what was the temperature you have used like for cross-linking, and have you used any slicing column after cross-linking?
Yes, so we are cross-linking on ice at the moment. We basically did a whole screen of cross-linking conditions, various concentrations, but all on ice, but for varying times. And 40 minutes on ice at 0.2% glutaraldehyde is basically the point where we see mostly cross-linking to dimers without seeing anything higher order. And so we went with this, and always after cross-linking, we run size exclusion. So we quench this with a tris buffer, and then we run it on size exclusion right after. And we do not see any differences between cross-linked and non-cross-linked complexes, which is why I didn't show it. But if you showed me a HER2, HER3 size exclusion profile, I could not tell the difference between something that's cross-linked and something that's not. However, our negative stain looks a lot better. And the Cryo-EM seems to be improved as well with the cross-linking. The other question on GO thickness, short answer, like we almost certainly in some areas have multiple layers of GO. Whether this limited our structure, our resolution to 3 angstrom, I don't know. We did not pay much attention to whether we should only include particles that come from single layer of graphene oxide, because it's just very hard, I guess, practically to do that. If we take GO grids that have 2 thin GO layers, then we can throw out more than half of our data sets, which is the other good thing is about making these in-house is that we can use slightly thicker graphene oxide. It doesn't seem to interfere much with the resolution, but you're very right. I would stay away from those areas where it's very, very thick GO. And then also you get overlapping particles and you can't really pick individual particles anymore.
Yeah.
Yeah.
Thanks
So
for
it's
that.
a little bit of a, there's upsides and downsides to both. Sometimes we get data sets that are terrible. Believe that it's not the sample, it can be the GO sometimes. It's very hard to say.
Yeah.
But there's definitely more variability with GO, and having a good batch of grids probably doesn't hurt.
Yeah. Thanks for that. Thanks for answer. Appreciate.
Yeah. I think we are running a little bit over time. Raphael, is it okay for you to stay a little bit longer to attend the questions?
Yeah, for sure.
So, Michael, can you please do yourself and ask the question?
Yeah, thanks. Great talk, Raph. Really nice. My question is just about the tip that you suggested about using kinase inhibitors to help purify the protein and complex. Are there any particular requirements of what you would select for the kinase inhibitor that you're using for this?
Yeah, so the kinase inhibitor we are using, if we can, then we use covalent kinase inhibitors. So the one for HER2 is called canadenib, and it's a covalent inhibitor. So we basically have to add it once just to the expression cultures, and then inhibits the kinase. Obviously, if you don't activate them, there's less downregulation of the receptor, which is why I think we get good yields. And then we usually don't add any kinase inhibitors at any stage downstream, because it should be bound, and we use like a lot of kinase inhibitor. So it should be in there. We also have, for HER3, there's another one, which is just a competitive inhibitor. It does also make a difference for expression level on HER3, but in that case, we would add the inhibitor again later before these grids, or sometimes even during purification in certain stages. So this is maybe not perfectly optimized, but we tend to use covalent inhibitors if we can, because it just makes it simple. If you don't have them, I would probably keep other inhibitors around, but putting it into the expression culture can help.
Great,
and it changes your phosphorylation state of the kinase too. So it can help or can be bad, but something to screen for sure.
Yeah, it's almost about 11 for those who have to leave right now. Thank you all for attending. And please note that our next seminar will be on June 14th. Also, thank you, Dr. Trenker. Yeah, let's quickly move on to our next question. Pramod Kumar has some questions in the chat box. Pramod, do you want to unmute yourself and ask your question? Hello? Okay. Can we move on to Isabel's question?
Just to follow up on Michael's question, Raph. So the kinase inhibitors, depending on which type you're going to use, is going to impact on the conformation and whether or not you stabilize or destabilize the complex, I would think. So have you screened a bit, because you can make the complex of the intracellular complexes? Have you screened, or are you doing that? Screening for small molecule or testing the compound that will stabilize or destabilize the complex?
I don't do it on just the isolated kinases, you mean?
Yeah, because you could actually use that to screen for stabilizer, or you know what I mean, because the conformation of the HER3 will impact on the conformation of the HER2, and then will impact. So it's good to do with stabilization of the alpha C helix. So it might be a way, I don't know, to screen.
It's a very good point. We have lots of inhibitors. We haven't tried them. I mean, we do know of a lot of inhibitors that stabilize the individual kinases. We haven't really measured much the HER2-HER3 complex. It's also a difficult one to form. if we choose them, we tend to, so Cardiovascular, for instance, was one, like if we choose type 1, we just assume that a type 1 inhibitor would be the right way to go with because it stabilizes the active conformation that you would expect to have in this heterocomplex. And this seemed to work out well. There's a couple of type 2. I've only tried it on the full-length proteins. It's always been difficult.
Okay.
That I can say. But for me, I haven't purified a kinase in a while, to be honest. But it's a good way to think, to maybe screen it. You're very right.
Yeah.
For me, it's almost faster to just purify the full-length receptors and then screen by negative stain EM. But yeah, you're right. We could use some more some more screening method to actually try and see. But I have to convince myself first that it's a kinase problem. I expect the reason we don't see them is not because of the kinases being unhappy. I just suspect it's something else. And I think going into either, I mean, it's very much future and I will probably not be doing this, but looking at the receptors in a cell membrane, like a real environment, reconstituting into liposomes and having some even downstream proteins bound to just add some meat to the intracellular domains, if you will.
All right, thanks.
Yeah, Pramod Kumar, please ask
Yes.
the question.
Hi, Raphael, wonderful talk. So I was wondering, is there any interaction between the transmembrane region of HER2 and HER3 complex? And are there any known mutation which affects the stability of the complex from the transmembrane region?
Yes, there are. Okay, so there's various mutations that sit in transmembrane and juxtamembrane regions in HER2. Presumably they work together with HER3. We have made them in the past. I haven't gone back lately. Ever since we got a good handle on these things, I actually haven't tried the transmembrane domain mutations again. But yes, so we do think that the transmembrane domain interacts via glycine XX glycine motifs, like a lot of other transmembrane helices, like the glycophorin A typical motif as well. All 4 HER receptors have them. And this is probably what happens in the transmembrane domain. And there are different structures. These are for EGFR, where upon activation, the transmembrane helices switch from having a dimer held together at the bottom and at the top. So there are ways of doing that. Yes, I haven't gotten around to do it, but it's probably something we should be doing, because what we really want to restrict is probably the relative movement of transmembrane to ECD and kinase. So fishing for some mutations in the juxtamembrane kinase region, transmembrane region will probably be a good idea. Yeah, you're very right.
I have another question. So you have tried nanodisk. Which MSP are you using for the nanodisk? And so are you like in the process of doing it? And what is the reason it's affecting the resolution in the nanodisk, in your opinion?
Yeah, so we have not put HER2-HER3 in the nanodiscs. We have another HER receptor that's in the nanodiscs, where I optimize it to a way that we can get good nanodisk reconstitution. At the moment, it works pretty good with MSP1E1. So like it's on the smaller side. We've also tried larger ones. For me at the moment, the bottleneck has been getting good particle density on a cryo-EM grid with these nanodiscs. We've tried various GO grids. We don't have enough concentration to see anything on just quantifiable wholly carbon grids. So I think figuring out how to reliably concentrate on nanodisk sample and freezing it is currently our bottleneck. So unfortunately, I can't quite tell you yet whether it will help our kinases. There's a paper by John Currien's group from Berkeley. They incorporated EGFR dimers into nanodiscs. I forget which nanodisk it is, but they have even similar problems where they're not able to resolve the kinases even in the nanodiscs. So that was a bit discouraging, to be honest, but we'll still do it and see if we can do a little bit better. But yeah, it's more on the EM issue at the moment rather than incorporation.
Thank you. I have another question. So did you had any problem expressing the full length protein in the beginning in mammalian cell?
Yes, so in just adherent 293s, there's always some protein, but always just very, very little. But I think this is probably just because the XP cultures in a 3-dimensional culture, you just get so much more cells out of it that it just made it so much easier. So we basically could replace 30 10-centimeter dishes of adherent cells by a 30 milliliter XP culture. So I can't really speculate too much on why adherent cell culture wasn't good enough, but I assume it's just expression is always low and you really want a lot of cells, would be my guess. And the XP cells are just very good at pumping out receptors. And
so how much of cells you started used for the suspension to get the protein, enough protein on the grid?
Out of 120 milliliter, we get enough to freeze 12 EM grids easily. So we usually usually grow 30 milliliter cultures, and using 4 of these 30 milliliter cultures gets us there. We usually also express the proteins individually, so I can mix and match different mutations of HER2 with HER3 and mix the cell lysates. But yeah, 4 30 milliliter cultures is usually enough for us to get a signal on size exclusion. And if you have a signal on size exclusion, you can usually get enough particles on a GO grid.
That's
But
good. Thank you very much.
Okay. Yeah, sorry. Can we move on to our next question? It's from Marielena. Marielena, please unmute yourself and ask your question.
Yeah, hello everyone. Thank you, Dr. Trenker. You gave us an amazing talk, and thank you also for being so generous to share so many tips with us. So I was wondering, how conserved is the dimerization arm of HER2? And have you seen in the literature any mutation there? And if yes, how does that affect the formation of dimers, their stability, etc.?
Yeah, that's a good question. We looked at conservation of the HER2 dimerization arm within the family, so the HER2 dimerization arm is, let's say, somewhat conserved. About, I have this figure somewhere, but there's a lot of residues there that are conserved within the entire family. Over evolution, it's pretty well conserved. We could not find any, at least not any oncogenic mutations that stand out to us that are in the dimerization arm. So we've looked very hard at the dimerization arm to see if there's something obvious in terms of sequence that could tell us whether it's stabilized or not. We can't really find very much that's obvious. And our current bet is that dimerization arm binding is more determined by the ligand that is bound to the dimerization partner than the actual sequence, but we might be wrong. We don't know for sure, but so far all the information that we have point towards the connection of ligand binding to dimerization arm stabilization. And I guess we need a couple more structures to see if that is truly the case. But this is what I think the current structures are most aligned with. And we have some other structures. I didn't really talk about it, but there's other crystal structures of EGFR where the structures are asymmetric and the dimerization arms are different. And we see that one dimerization arm, even in EGFR, has higher B factors than the other one, indicating that there's just more movement on the side where the ligand is not very tightly bound. So all indications point towards the ligand connection, but remains to be seen, I guess. But yeah, it's a good question, and we're always out for finding some mutations that change the way the dimerization arm works. Thanks.
Thanks so much,
Yeah.
Anjan. Great talk.
Thanks.
Yeah, that was our last question. Thank you, Dr. Raphael. for staying this long. And thank you all for attending this seminar. Hope to see you in our next seminar. Yeah.
Awesome. Great meeting you, and thanks for having me.
Yeah.